Among the most enigmatic and often overlooked contributors to vitamin B12 deficiency are rare genetic mutations that impair its transport, absorption, or intracellular metabolism. These are not the conventional cases of low dietary intake or malabsorption due to aging or gastrointestinal disease. Instead, they represent a silent biochemical miscommunication where the body may receive B12 yet fail to use it properly. In such individuals, functional B12 deficiency develops despite apparently normal blood levels, creating a diagnostic gray zone where symptoms may be profound but lab results remain misleading. This genetic dimension transforms B12 from a simple nutrient into a complex, individualized cofactor whose effectiveness hinges on an invisible architecture of inherited molecular pathways. One of the most studied mutations in this context is the MTHFR, methylene tetrahydrofolate reductase, polymorphism, particularly the C677T and A1298C variants. While technically a folate cycle gene, MTHFR plays a crucial role in the remethylation of homocysteine, a process dependent on both B12 and folate. Individuals with these variants often exhibit impaired methylation and elevated homocysteine, even when B12 and folate intake are adequate. For them, the need for methyl donors, including methylcobalamin, the active form of B12, may be significantly increased. They are not merely B12 deficient in the classic sense. Rather, their biochemical machinery is wired in such a way that standard doses may be insufficient to meet functional needs. Yet other mutations lie deeper in the B12 pathway itself. For example, the TCN2 gene, which encodes transcobalamin 2, is responsible for binding B12 in the bloodstream and delivering it to cells. A common polymorphism in this gene, TCN2C776G, can reduce the efficiency of B12 transport into tissues, leading to intracellular deficiency even when serum levels appear normal. People with this variant may show signs of neurological decline, fatigue, and impaired cognitive function, none of which improve unless higher doses of active B12 are administered, bypassing the defective transport mechanism. In this scenario, the bloodstream becomes a deceptive reservoir. B12 is present, but cells are starving. Even more rare are mutations in genes like MMACHC, which affect the intracellular conversion of B12 into its coenzyme forms, methylcobalamin and adenosylcobalamin. These defects, found in inherited disorders like CBLC or CBLD types of methylmalonic acidemia and homocystinuria, illustrate just how specialized and tightly controlled B12 metabolism is at the cellular level. While such cases are usually identified in infancy or early childhood, milder polymorphisms in these pathways may persist unnoticed into adulthood, causing chronic issues such as developmental delays, psychiatric symptoms, and metabolic disturbances. Because these conditions often evade standard blood tests, they require more sensitive biomarkers like methylmalonic acid, MMA, or homocysteine, which reflect cellular utilization rather than mere serum concentration. This genetic backdrop reshapes our understanding of what it means to be B12 deficient. It is not always about how much B12 is consumed, but whether it can be effectively internalized, activated, and used in key metabolic reactions. For those with polymorphisms in B12, binding proteins, methylation enzymes, or mitochondrial cofactors, functional deficiency can manifest in ways that look like mood disorders, fibromyalgia, chronic fatigue, or even early cognitive decline. These symptoms may persist for years, mistakenly treated as psychiatric or stress-related, until a deeper biochemical assessment uncovers the hidden genetic mismatch. The good news is that these genetically influenced inefficiencies can often be managed with targeted nutritional support. Methylcobalamin and adenosylcobalamin, the active forms of B12, can bypass many enzymatic bottlenecks and deliver coenzyme activity directly where it is needed. In some cases, high-dose hydroxycobalamin is preferred, especially in individuals with nitric oxide-related oxidative stress. For those with impaired folate, B12 synergy, adding methylfolate can further enhance the methylation cycle, optimizing detoxification, neurotransmitter production, and DNA repair. The key is personalization, moving beyond generic RDA levels and tailoring B12 therapy to match the unique genetic and biochemical context of each individual. Ultimately, the presence of these mutations invites a paradigm shift, from seeing B12 deficiency as a binary state to recognizing it as a spectrum of functional inefficiencies. This model explains why some people thrive on standard multivitamins, while others need aggressive, high-dose intervention. It also suggests that so-called idiopathic cases of depression, brain fog, or chronic pain may be rooted not in psychological weakness, but in a subtle, inherited fragility of B12 metabolism. In this way, B12 becomes more than a nutrient. It becomes a variable in a person's biochemical identity. And for those living with unexplained fatigue, mood swings, or neurological instability, these rare mutations may hold the key not only to diagnosis, but to transformation. By unveiling the genetic architecture behind B12 utilization, we open a window into truly personalized medicine, where treatment is not just about correcting a lab number, but about restoring the silent cellular conversations that sustain life and vitality.